Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. In this short episode, we are focusing on a crucial topic for our higher biology students, but also for anyone interested in the wonders of cellular biology, DNA replication. Before any cell divides, it's got a monumental task: to copy its DNA precisely. This ensures that each new daughter cell receives a complete set of genetic instructions from the parent cell. But how does that complex process work? Let's break it down. Firstly, DNA replication isn't a haphazard process. It requires certain things, including a DNA template. Think of that as the original blueprint. A supply of free DNA nucleotides. These are the building blocks, namely adenine, thymine, cytosine, and guanine. It needs a primer. That's the starting point for the new DNA strand. It needs ATP, because nothing happens without energy, and enzymes, specifically DNA polymerase and DNA ligase, which play pivotal roles. The next thing you need to know is an overview of the stages of DNA replication. Firstly, unwinding the DNA. The enzyme DNA helicase unwinds the DNA's double helix, separating the two strands by breaking the hydrogen bonds between the bases. Secondly, the role of the primer. DNA polymerase can't just start adding nucleotides willy-nilly. It needs a primer to latch onto the 3' end of the DNA strand. And thirdly, building the new strands. DNA polymerase adds complementary nucleotides to the growing DNA strand. The replication on the leading strand is continuous, while the lagging strand is synthesized in fragments. So why is this DNA replication so important? Simply put, DNA replication is vital for life. It ensures that each new cell has the same genetic information as the parent cell, essential for growth, development, and maintaining the organism's integrity. Now let's talk about a powerful technique used in laboratories worldwide, the polymerase chain reaction, or PCR. PCR allows scientists to amplify a specific DNA segment from a small sample to millions of copies. This is crucial in various fields, including forensic science, genetic testing, and medical diagnostics. PCR involves the use of a PCR machine known as a thermocycler. Other requirements for PCR include primers, heat-tolerant DNA polymerase, a supply of 4 types of DNA nucleotides, and a DNA template, the piece of DNA that needs to be amplified. And again, we need to know the stages of PCR. So firstly, the DNA molecule is denatured, where it's separated into 2 single strands by heating to 95 degrees centigrade for a few seconds. Secondly, it's cooled down to 50 to 65 degrees centigrade to allow primers to bind to target sequences. And thirdly, it's heated again to allow the heat-tolerant DNA polymerase to add nucleotides to the 3' end of the new DNA strand. The DNA molecules are then heated to 95 degrees centigrade again as we go back to step 1 to separate the strands, and the process is repeated. Each cycle takes about 20 minutes, with the DNA content doubling each time. PCR has revolutionised many aspects of science and medicine, including forensics, solving crimes by amplifying DNA from crime scenes, parentage disputes, identifying biological relationships, and diagnosing genetic disorders, detecting and understanding genetic diseases. Understanding DNA replication and techniques like PCR opens up a world of possibilities in biology, medicine, and beyond. Understanding these concepts not only helps us prepare for our exams, but also for a future in the fascinating field of biology. So hit subscribe on whatever platform you get your podcasts, so you don't miss the rest of our short summary episodes, nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow, such as Scholar or WestOS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website, www.leckyscotland.co.uk.